Hello students, welcome to Saurasya classes. Today our topic is cells and development, and in this video I'm going to discuss about this statement, cell, the basic unit of life, and I'm going to discuss it through some points. So now let's get started. Okay, so my first point is Robert Hooke coined the term cell. For small structures in a piece of cork under a microscope. So this was for small structures structures in a piece of cork under a microscope in 1665. His observations were published in his book Micrographia. So his observations were published in his book Micrographia. Graphia. So the first point was Robert Hooke, who coined the term cell for small structures in a piece of cork under a microscope in 1665. His observations were published in his book Micrographia. Now the next point is Leeuwenhoek. The next one is Leeuwenhoek. In 1674, that means after Robert Hooke, he observed one-celled structure like bacteria, protozoans, and blood cells. Now, later it was confirmed that plants were also made up of cells. Made up of cells. So, second one is Leeuwenhoek in 1674 observed one-celled structures. One-celled structures like bacteria, protozoans and blood cells. Later, it was confirmed that not only animals but also plants were made up of cells. Now, coming to a third point, that is, cell is now defined as structural and functional unit of the body. Cell is now defined as structural and functional unit of body. There are two general classes of cells. Now, we can divide cell in two general classes. Cell Now, the one is prokaryotic cells. First one is prokaryotic cells, and the second one is eukaryotic cells. Eukaryotic cells. So, cell can be defined as the structural and functional unit of body, and we can again divide cell in two parts that are prokaryotic cells and eukaryotic cells. Now, our next point is about cell size. Cell size. Okay. Now, two important factors contribute to the cell size are the two important factors that contribute to the cell size are, the first one is nucleocytoplasmic ratio. Okay, let me change the color. Okay, the first one is nucleocytoplasmic ratio, and the second one is surface area of the cell. Surface area of the cell. So, the size of the cell depends on two factors, and these are nucleocytoplasmic ratio and the surface area of the cell. Now, the next point is cytology. Now, there are again some features and informations about cytology, and let's discuss in points. The first one is colloidal theory. Colloidal theory. Protoplasm is polyphasic colloidal solution that generates energy of life. So the colloidal theory states that protoplasm is polyphasic colloidal solution polyphasic colloidal solution that generates energy of life, that generates energy of life. That was colloidal theory. Now the next one is protoplasm theory. Protoplasm theory. The protoplasm theory states that an organism is made up of protoplasm, and cells are simply an accumulation of protoplasm. So it states that an organism made up of protoplasm and cells are simply an accumulation of protoplasm. So the protoplasm theory states that an organism that is made up of protoplasm and cells are simply the accumulation of protoplasm. Now the next we are going to discuss is the organismal theory. Let's change the color and take a new one. Okay, organismal theory. No, it's very okay. Take this one. Organismal theory. So according to the organismal theory, an organism is a continuous mass of protoplasm segmented into cells. So according to this theory, an organism is a continuous mass of protoplasm is a continuous mass of protoplasm and that is segmented into cells. Segmented into cells. Idea of cell theory was first given by the idea of cell theory was first given by Wbjee Aiims in 1824. So, according to this organismal theory, an organism that is a continuous mass. An organism is a continuous mass of protoplasm and that is segmented into the cells. And the idea was first generated in 1824. So, now let's come to the cellular totipotency. The next point we are going to discuss is cellular totipotency. Okay, again I'm going to discuss this through some points, and the first one is the capacity of a cell to form whole of the organism is called the totipotency. So the totipotency is the capacity of cells to form whole of the organism. The organism is called the totipotency. So the capacity of a cell to form the whole of an organism is called the totipotency, as called by Morgan. This was stated by Morgan. Now the next one is pluripotency. That means the next term we are going to discuss is pluripotency. Pluripotency means the ability to de-differentiate completely after the cell maturity. So pluripotency means the ability to de-differentiate differentiate completely after cell maturity. After cell maturity. So, pluripotency is the capability to dedifferentiate completely when the cells are mature. The next one is the next point is the German botanist Gottlieb was the first to culture plant tissues in vitro. So, German botanist Gottlieb was the first to culture plant tissues in vitro. Now, the next is callus is the mass of undifferentiated cells formed by growth of a totipotent cell in culture. So, callus is the mass of undifferentiated cells formed by growth of a totipotent cell, which is formed by the growth of a totipotent cell in culture. So, callus is the mass of undifferentiated cells. It is the mass of undifferentiated cells formed by the growth of a totipotent cell in culture. Now, the last point is callus was first time differentiated to whole plant of tobacco by Hooke and Miller. So, callus was first differentiated to whole plant of tobacco by Hooke and Miller. So, the term callus was first differentiated to whole plant of tobacco by Skoog and Miller. So in this video I have discussed some points through which we can just support the statement that was sale, okay, that was sale, the basic unit of life, and I have discussed these points. Thank you.